Good morning. Hello, everybody. All the agriculture researchers always facing with this question: what is a quick solution for crop grain biofortification? I am Shahram Mahmoud Soltani, Assistant Professor in Soil Chemistry and Plant Nutrition from Rice Research Institute of Iran. I'm going to discuss and share my last research finding entitled Zinc and Iron Biofortification and Enhanced Protein Content of Rice in comparison between zinc and iron chelated with glycine amino acid versus zinc sulfate. Of course, all of you know rice is a strategic and leading staple food crop for more than 50% of the world population. Although polished rice provides 15% of protein and 20% of dietary energy per capita, however, removal of the nutrient-rich aileron layer and embryo make it a poor source of important and essential micronutrients. On the other hand, more than 25% of the people in rice growing area suffer from this problem due to lack of zinc and iron in their white rice. But what is the solution? Biofortification is a sustainable and a quick solution for overcome this problem. Biofortification is the process by which the nutritional quality of food crops is improved through agronomic practices, conventional plant breeding, or modern biotechnology. A structured, effective agronomic strategy are widely used to increase zinc and iron soil availability and consequently their phytoavailability for optimum crop growth and development, and more broadly, biofortification in grain. The backbone of all these strategies rely on different sources, rate, and application method of this micronutrient fertilizer. Of course, many field researchers show the fact that the zinc and iron amino acid chelates compared to other products indicated the more effective performance on not only rice morphological trait, yield, and yield component, but also their nutritional quality, particularly protein, zinc, and iron content of head rice. Therefore, we set an experiment with this objective. The first, to evaluate the effect of the foliar application of iron and zinc chelated with glycine amino acid and zinc sulfide fertilizer on the rice nutritional quality. And second, to compare the effects of amino acid chelate with sulfide fertilizer on above mentioned traits. A field experiment was conducted on the research field of the Rice Research Institute of Iran at Rasht, Iran during rice growing season of 2019 and 2020. The 12 experiment treatments were conducted in a randomized complete block design with 3 replications. Rice seedlings of the most common Iranian local rice cultivar, Hashemi, were transplanted in all pots, 3 seedlings per hill in a 2020 pattern. The applied treatment were T1 or control. Also, we applied recommended rate of macronutrient fertilizer. T2, T3, and T4 is T1 plus foliar application of 1.5 kilograms zinc per hectare in source of zinc chelated with glycine amino acid. T5, T6, and T7 is equal to T1 plus foliar application of 1.5 kilograms zinc per hectare in source of zinc sulfate. T8, T9, and T10 is equal to T1 plus 1.5 kilogram iron per hectare in the form of iron chelated with glycine amino acid. All zinc and iron chelate of glycine amino acid manufactured by Pagostar company in Iran. All applied treatments except T1 were applied at the booting and ripening stage. 50% of experimental rates for each stage applied on each rice growth stage. The zinc and iron glycine amino acids contain 26% zinc, 20% iron, and between 27 to 30% glycine amino acids. The pictures show how we apply the applied treatment foliarly in the booting and ripening stage. And we measure zinc in grain and white rice, iron in grain and white rice, and protein content in white rice. We measure zinc and iron in rice tissue by using the dry ash method. And the total protein content of white rice that we measure using the soil and water research institute method. In this method, total N content multiplied is 0.85%. The coefficient for local RAS changing the total N content to protein content. The ANOVA result, especially in the second row, showed all applied treatment positively significantly influence the measured traits at 1% confidence level. For zinc concentration of rice grain, the figure show if you follow the horizontal line, all applied treatments positively significantly increase the zinc concentration of rice grain. But by application of zinc glycine amino acid, the green arrow, compared to the red arrow, the control, the result indicated 25.5% increase compared with the control. But when we compare the zinc amino acid chelate compared with the zinc sulfate, the blue arrow for zinc sulfate indicated zinc amino acid chelate is 7% more effective than the zinc in source of sulfate. For zinc concentration of white rice, with the same train with the rice grain, all applied treatments significantly increase the zinc concentration of white rice. You can look at the horizontal line in the figure. But zinc amino acid chelate increased 20% more than the control. By comparing the green arrow for zinc amino acid chelate compared the red one, the control. But in comparison between the zinc amino acid chelate, green one, with the zinc sulfate, the blue one, the result indicated 6% increase in zinc concentration of white rice through application of zinc in the form of chelated with the glycine. For iron concentration of rice grain, still almost all of applied treatments, especially iron amino acid chelate, increase the iron concentration of rice grain, but iron chelated with the glycine amino acid increased 2.2 times in compared with the control in iron concentration of rice grain. Also, the interesting thing is the application of the zinc chelated with amino acid chelate, the blue arrow. We received 25% increase in iron concentration of rice grain compared with the control. And in compared with the zinc chelated with amino acid, and zinc sulfate. The blue is the result indicated 10%. The zinc amino acid, zinc glycolated amino acid is more effective than zinc sulfate. For iron concentration of white rice with same trend, iron glycine amino acid increases 15.5% more than the control when you compare the green arrow with the red arrow. Also, zinc amino acid increased 50% in iron concentration of white rice in compared with the control. The blue arrow compared with the red arrow. And also, in compared with the zinc with different source, the zinc glycine amino acid increased as same as zinc sulfate. It means for iron concentration of the white rice, we cannot find significantly different between the zinc glycine amino acid and zinc sulfate. For protein content of white rice, still almost all of the treatments increase the protein content of white rice, but the zinc sources are more effective than iron in the form of glycine chelate. But zinc glycine amino acid chelate increases 10% more than the control with no application, any full application of the zinc. And in compared with the zinc glycine amino acid and zinc sulfate, only 1% zinc amino acid chelate is more effective than zinc sulfate. Therefore, despite of sources and rates, zinc and iron application enhance all rice nutritional quality traits. But unfortunately, milling process grain to white rice significantly decrease the rice nutritional quality traits. And finally, we can conclude that the iron and zinc glycine chelate increase the rice nutritional quality traits more than the zinc sulfate. At the end of my presentation, I would like to express my special gratitude to Pagbo Star Company for financial support of core research, to ES 2022 conference organizer for giving us the oral presentation opportunity, and to Academy of Excellence in Bioscience for their kindly sponsoring for registration. Thank you for your attention. And for more information, please contact my email. Thank you and goodbye.